Hey everyone, this is the last short lecture for the first day of class this quarter. Just wanted to talk a little bit about um, microscopy, and this is a bit of a lead in into lab this week also. So let's just look at our friend Anton van Leeuwenhoek, a Dutch businessman, sort of um, citizen scientist back in the 1600s, who was really the first microbiologist in the sense that he built his own microscopes. As you can see here, these are kind of amazing little devices that had a ground glass lens, magnifying lens here. And right here on this little tip is where he would put his specimens, including, for example, drops of pond water or scrapings off his teeth or whatever he wanted to look at. And he actually was the first person to be able to see the invisible world of microorganisms. He created microscopes, these simple microscopes that could magnify up to 300-fold, which is actually enough to see large bacteria. And he actually drew these. You know, they look like bacteria and protozoa and many other types of unicellular and small multicellular organisms. He described them in dozens of letters that he published with the Royal Society. So he was truly the first microbiologist actually seeing and describing microorganisms. Amazingly, things didn't really change much for a few hundred years after van Leeuwenhoek into the 19th century. Microscopes got bigger and easier to use, but the magnification and resolution didn't change that much. And if you're wondering what resolution is, first I'm going to say I want you to read Appendix B in your textbook, and I also have a link to another great resource on microscopy on the lab worksheet for this week. But basically, resolution is how far apart things can be, and you can still, or how close together they can be, and you can tell that they're actually separate. So what's sort of, you can think of it as the level of detail that you can see in your image. And, you know, so van Leeuwenhoek's microscope was what we call a bright field microscope. It just uses regular visible light. And, you know, things didn't change that much for quite a while. Dark field microscopy, which you can look at in Appendix B, was invented in the 1830s, but really it wasn't until like the last century that many real advances in the technology of microscopes came along. So I just have a list of them here. These are all described in Appendix B. I would like you to learn what all these kinds of microscopy do and what they can be used for in microbiology, because we are going to be looking at images from many of these over the course of the next few weeks. And it's important to understand what type of microscopy can do what tasks for you in microbiology. But I'm not going to go over them all now. Again, I want you to read that section of your textbook. All right, so here's sort of a depiction of the microscopic world and the sort of the size ranges that we're talking about. And I do want you to have sort of a feeling for this. The limit that the human eye can see is down to about 1/10 of a millimeter, right? 100 micrometers. And I want you to make sure you understand this technology too, right? So a millimeter is 1 times 10 to the minus third meters. A micrometer is 1 times 10 to the minus 6 meters. And a nanometer is 1 times 10 to the 9th meters. All right, so this is our human eye range down to a little below the millimeter here. So anything below this is microscopic. Now, a light microscope will take you down to about 0.2 microns, right? So about 3 more orders of magnitude. in the size range, right? And this encompasses bacterial cells and eukaryotic cells. Eukaryotic cells are typically significantly larger than bacterial cells. Average bacterial cell might be 1 micron in length or diameter. Some of them are bigger, some of them are smaller, but the limit of size of a bacterial cell is about 0.2 microns. And then in that range, now you're starting to get into the range of viruses. So viruses go down to about maybe 10 nanometers would be a very small virus, because now you're getting into the size range of macromolecular complexes like ribosomes. And then down below that, you're getting into the range of individual proteins and ultimately down into the atomic range. So an angstrom is 1/10 of a nanometer, right? Okay, so below 0.2 microns, what you really need is an electron microscope. There are applications for electron microscopes that are a lot bigger than that, especially scanning electron microscopes. But below the range of a light microscope, you need to use an electron microscope. Okay, so all this is, again, is talked about in Appendix B. So I want you to make sure you go through that. Now there are exceptions to the range that I just told you about, right? There are actually some bacteria, super large bacteria, that go up to almost a millimeter in diameter. Here are a couple of interesting examples that have been discovered in the last decade or so. We'll talk more about these, maybe a few examples of these later on, because they're really interesting bacteria. At the lower size range, we have the mycoplasma. These are bacteria that are only about 0.2 microns in diameter. And here's a question that I would like you to actually think about, and we'll talk about, maybe do some discussion in class. What factors actually limit the upper and lower sizes of bacterial cells? Why is it so rare to have a bacteria this big? Why is it that we don't have bacteria that are actually smaller than this, right? We'll talk about that in class. Okay, last thing I want to say before coming to lab this week, I want you to make sure you've read Appendix B about microscopy. And I also want you to at least look through the protocol. In this case, it's a worksheet for week 1 before you actually come to lab. You're going to be doing it during lab. It's available now on Camino, and so I want you to take a look at that. Before Thursday's class, I want you to read through, or at least scan through chapter 1. These are the things we talked about today. Although I won't be, and this is actually what's going to be on the quiz. This and Appendix B are going to be on the quiz that you need to do by Friday night. For Thursday's class, I want you to have read the start of chapter 2, sections 2.1 to 2.3. All right? I will see you soon then. Thanks for watching.